Hello everyone, this is the first part of the cardiovascular system, namely blood. Blood is a connective tissue. It functions distributing substances such as delivering oxygen and nutrients to body cells, transporting metabolic waste to lungs and kidney for elimination, transporting hormones from endocrine organ to target organ. Also, the function is regulating blood levels of substances for maintaining body temperature, normal pH, and fluid volume in circulatory system, and also protective function for preventing blood loss by plasma proteins and platelet by clot formation, and also preventing infection. When you draw the blood and then spin, it yields the top layer as a plasma, about 55% of volume, and uh, red blood cells are on the bottom, about 45%, and in the middle, less than 1%, bumpy coat contains the white blood cells and the platelet. Uh, Blood plasma contains mostly water, 90%, and contains more than 100 dissolved solutes. Solute contains nutrients, gases, hormones, waste, proteins, inorganic ions. The plasma proteins are made by liver, and those are albumin, globulin, and fibrinogen. And albumin is very important substances carrier and blood buffer, and also major contributor of plasma osmotic pressure. Formed elements as shown here. White blood cells are complete cells, and red cells are in the background, has no nucleus or organelles, and plates are cell fragment. Red blood cells has a structural characteristic to contribute gas exchange or transport, and their shape is a biconcave shape, shows a huge surface area relative to volume, and they are filled with hemoglobin for gas transport, and also contain a special protein on the cell membrane called spectrin, and these provide the flexibility to change shape as red cells travel through capillaries. And also red cells are main factor contributing to blood viscosity. As you see, red cells has no nucleus, no organelles, so no mitochondria, so it produces ATP by cellular respiration and aerobically, therefore it doesn't consume oxygen, they only transport. Red blood cells binds, it has a hemoglobin that binds oxygen reversibly, and hemoglobins contain globin and heme, and globin has a 2 alpha 2 beta subunits, and each subunit binds oxygen, and each red blood cell contains almost 250 million hemoglobin molecules. Hemoglobin binds to oxygen in lungs, become oxyhemoglobin, and when it unloads oxygen in tissue, it produces the deoxyhemoglobin, and when it binds to the CO2, it becomes carbaminohemoglobin. Hemopoiesis is a blood cell formation, and normally hormones and growth factors push hematopoietic stem cells, which is hemocytoblast cells, to give rise to all formed elements. They first become committed cells, and then they go through developmental pathways to become mature red blood cells. During the developmental pathway, ribosomes are synthesized, hemoglobins are synthesized, and then ejection of nucleus and formation of reticulocytes, which is young red blood cells, occur. When reticulocyte ribosomes are degraded, then they become mature erythrocyte in 2 days after entering blood circulation. Therefore, reticulocyte counts indicate rate of red cell formation. In human body, normally almost 2 million red blood cells made per second. Underproduction of red cells lead to tissue hypoxia, or overproduction of red blood cells increases blood viscosity. Therefore, regulation of balance between red cell production and destruction depends on hormonal control, adequate supply of iron, amino acids, and B vitamins. Hormone EPO, erythropoietin, is a direct stimulus for red cell production. EPO always exists small amounts in blood to maintain basal rate of the red cell production. High red blood cell or oxygen levels depress its production, and it is released by kidney, some from liver, in response to hypoxia. Dialysis patients normally indicating kidney problems have a low red blood cell counts. Factors triggering hormonal control of erythropoiesis are hypoxia and decreased red blood cell number due to hemorrhage or increased destruction, or insufficient hemoglobin per red blood cell, such as seen in iron deficiency, and reduced availability of oxygen by going into high altitude. These factors trigger kidney to make more EPO, and more EPO can be more committed marrow cells and then more reticulocytes. And also, testosterone has shown to enhance EPO production, resulting in higher red blood cell count in males. Um, the fate and destruction of erythrocytes are seen in this slide. Um, Once new red blood cell enter bloodstream, it functions about 120 days. During this lifespan, no protein synthesis, growth, or cell division can occur. At the end of lifespan, old red blood cells become very weak, and hemoglobin start to degenerate and get trapped in small circulatory channels, especially in spleen, where macrophages engulf dying red blood cells. Heme and globin are separated from the hemoglobin, and ions are salvaged for reuse. Storage form can be ferritin or hemosiderin, and transport form can be transferrin. Heme are degraded into yellow pigment called bilirubin, and liver secretes bilirubin in a form of bile into intestine, where it can be degraded into urobilinogen, which is shown in feces as a steatobilin. Globin is metabolized into amino acid and are released into circulation. Anemia is an erythrocyte disorder. Blood has abnormally low oxygen carrying capacity. Blood oxygen levels cannot support normal metabolism. Therefore, patients are accompanied with fatigue, pallor, shortness of breath, and chills. So, cause of anemia can be by blood loss, low red blood cell production, or high red blood cell destruction. Sickle cell anemia is caused by high red blood cell destruction, and underlying problem is one amino acid mutation in a beta globin chain, and red blood cells are sickle shaped when oxygen level is low in the blood, or hemoglobin unloads oxygen. The result is that red blood cells rupture easily and block small vessels, creating poor oxygen delivery to that area, creating pain. Leukocytes, made up of less than 1% of total blood volume, their function is to defend against the disease. They can leave capillaries via diapidiasis and move through tissue space by anemoid motion and positive chemotaxis. There are two types of leukocytes, granulocytes and agranulocytes. Granulocytes has a visible cytoplasmic granules. Agranulocytes has a no visible cytoplasmic granules. And granulocytes examples are neutrophils, eosinophils, basophils. Agranulocytes example can be lymphocytes and monocytes. Neutrophils are most numerous white blood cells and have lilac staining granules, contain hydrolytic enzymes or defensins, have about 3 to 6 lobes in nucleus, and they are about 2 times bigger than red blood cells, and they are very phagocytic. Therefore, they are called bacterial slaters. Eosinophils show red staining granules and have bilobed nucleus and have lysome-like granules since they release enzymes to digest parasitic worms, and they have a role in allergies and asthma and modulating immune response. Basophils are rarest white blood cells and have nucleus deep purple with one and two constrictions and have a large purplish black granules which contain histamine. Histamines are inflammatory chemical that acts as a vasodilator to attract white blood cells to inflamed sites. Uh, these are functionally similar to mast cells. Agranulocytes, the lymphocytes, are second most numerous white blood cells and have a large dark purple circular nuclei with a thin edge of blue cytoplasm. They exist normally in lymphoid tissue, such as lymph node and spleen, and very few circulate in blood. They are crucial to immunity, and there are two types. T lymphocytes kills virus-infected cells and tumor cells, and B lymphocytes uh, produce antibodies. Monocytes are largest leukocytes and have abundant uh, pale blue cytoplasm and contain dark purple staining U or kidney-shaped nuclei. They leave circulation and enter tissues and differentiate into macrophages. They are actively phagocytic cells and they are very crucial against viruses, intracellular bacterial parasites, and chronic infections. And they activate lymphocytes to show immune response. Production of white blood cells is called leukopoiesis, which is stimulated by interleukins and colony stimulating factors. All leukocytes are originated from stem cells called hemocytoblasts. Lymphoid stem cells on your left become lymphocytes, and all others from myeloid stem cells on your left. Progression of all granulocytes seen on the left are from top to bottom: myeloblast, promyelocyte, myelocytes, band, mature cells such as eosinophils, basophils, and neutrophils. These granulocytes are stored in bone marrow and have a shorter lifespan and die by fighting bacteria. Progression of agranulocytes differ. Seen on your right, monocytes live several months and share common precursor with the neutrophils such as monoblast, monocytes, then become monocytes. Lymphocytes live few hours to decades. They are formed from lymphoid stem cell to T lymphocyte precursor and B lymphocyte precursors. Some of monocytes become macrophages, and some of the B lymphocytes become plasma cells, and some of the T lymphocytes become effector T cells. Platelet formation are made from megakaryocytes, and this process is regulated by uh, thrombopoietin. Megakaryocytes are large cells with a multi-lobed nucleus since they do mitosis without cytokinesis. Platelets are cytoplasmic fragments of megakaryocytes. They are made from blue staining outer area called purple granules. These granules contain an important molecules such as serotonin, calcium ion, enzymes, ADP, and platelet-derived growth factor, which acts in clotting process by forming temporary platelet plug that helps seal blood vessel damage. To prevent unnecessary clotting, circulating platelets kept inactive and mobile by nitric oxide, and they are from endothelial cell lining of the blood vessel. Hemostasis, a fast series of reactions for stoppage of the bleeding, requires clotting factors and substances released by platelet and injured tissue. 3 steps: vascular spasm, platelet plug formation, and coagulation. So here, vascular spasm uh, occurs when blood vessels cut. Vasoconstriction of the damaged blood vessel triggers direct injury to vascular smooth muscle, and then chemicals released by endothelial cells and platelets, uh, creating this vascular spasm. And this gives the pain reflexes, and this is very effective in smaller blood vessels. Hemostasis' second step is platelet plug formation, and this is positive feedback mechanism. And damaged endothelium exposes collagen fibers, and then platelet will stick to these collagen fibers, and as a result, platelet will start to swell and become spiky and sticky, and then they release the chemicals such as ADP cause more platelets to stick each other and release more of the chemical content such as serotonin, thromboxane A2, which enhance the vascular spasm and platelet aggregation. And third step is coagulation, which reinforces platelet plug with the fibrin thread, which leads to blood transformed from liquid form to gel form. This takes a series of reactions using clotting factors and also plasma proteins and vitamin K. Hemostasis as a coagulation phases first involves the prothrombinase formation in both the intrinsic and extrinsic pathways. So, and then prothrombinase will activate the prothrombin to thrombin, and then activated thrombin will convert fibrinogen to fibrin. So you need activation factors and also calcium, and at the fibrin mesh formation, it needs to be also strengthened and stabilize the clot, which require clot retraction. And here contraction pulls on fibrin strand and squeeze the serum from the clot and draws the ruptured blood vessels edges together to make a very stable clot, as you've seen this particular magnified figure. All right. ABO blood groups. You have 4 blood types: type A, B, AB, and O. Those are due to antigen agglutinin present on the red blood cell surface. And as you see there, type A contains A antigen, type B contains B antigen, type AB contains both A and B, type O has a no antigen. However, in a plasma, type A contains anti-B antibody, type P contains anti-A antibody, type AB has a no antibody, and type O contains both A and B antibody. So, anything perceived as a foreign generate the immune response by agglutination. That means red blood cells are clumped together. So mismatched transfused blood can be perceived as a foreign to be agglutinated and destroyed, and this can cause fatal situation. Therefore, presence or absence of each antigen is used to classify blood cells into different groups are very important for blood transfusion. And there are also other agglutinin, which is Rh uh, uh, antigen. So Rh plus means Rh antigen on the red cell surface. Rh minus means there is no Rh antigen on the red cell surface. So those antigen antibody, when they match, it creates clumping, and so that it causes a serious hemolytic reactions. First row shows the type AB antigens of red blood cell surface will react with anti-A and anti-B antibodies in the serum, creating clumping red blood cells and antibodies. Second row shows type B. Only anti-B serum reacts with the type B antigen on red blood cell surface. Third row shows type A. Only anti-A serum reacts with the type A antigen on red blood cell surface. Fourth row shows type O. No clumping occurs since no antigens are present on the red blood cell surface. Uh, think about which of the above is a universal donor and which of the above is a universal receiver.